All right, so we are continuing with our Unit 4, Lesson 15, which is on the nervous system. So last time we discussed homeostatic control. And we looked at how that worked through by looking at the endocrine system. And really, we looked at how under that system, the endocrine method of regulating and establishing balance requires some time. That imbalance has to first be detected, and then you have to have a stimulation of the appropriate glands. And once those glands are stimulated, they have to release their hormone. Then the hormone has to take some time to travel through the bloodstream to eventually reach its target cell and affect them and bring about the signal that's required. But you don't always have the luxury of having lots of time to respond to changes to external environment. Sometimes you have situations where the external environment changes very quickly. The organism has to respond very quickly to that situation. So the nervous system is able to respond to these types of quick changes within mere seconds or even fractions of a second. So here, today's lesson, we'll take a look at the structure of the nervous system, how it works, and how it brings about a response to changes to the external environment. And we will learn to discuss the nervous system through using the appropriate terminology related to the nervous system. We'll look at terms such as the synapse, acetylcholine, which is a neurotransmitter, look at the threshold in neurotransmission, the reflex arc, the axon, and the action potential. And as we're going through our lesson, we are going to be thinking about these questions. We're going to keep these on top of our mind as we're going through the lesson. Questions such as, do nerve cells carry any electrical current? Do some responses occur more quickly than others? How does the nervous system know what type of change in the external environment has taken place, and if a change in that external environment has taken place? And then how is the message then relayed to the brain? So the nervous system is a pretty elaborate and very complicated communication system that contains really more than 100 billion nerve cells. And it can be divided into main areas: the central nervous system and the peripheral nervous system. And we can look at that here. The central nervous system consists of the nerves of the brain and the spinal cord. And the central nervous system, the brain and the spinal cord, they are responsible for coordinating all those incoming and outgoing information and signals. The other part of the nervous system is the peripheral nervous system, which consists of the nerves that carry the information between all the other areas of the body and the central nervous system. And because of its complexity, the peripheral nervous system consists of nerves, can then be further divided into the somatic and the autonomic nerves. The somatic nerves are ones that control the skeletal muscles that are responsible for motion and motor. They are also responsible for the movement of skeletal muscles, the bones, the skin, which is sensory, and it relays the information about the external environment then back to the brain. And this part of the nervous system where we is one that where we have some measure of awareness and also some measure of voluntary control. We can sense the changes in the environment, such as heat, light, touch, taste, smell, and then we can respond to it by moving our bodies or laughing or smiling or those types of voluntary responses. The autonomic portion of the peripheral nervous system, this is not under our control. It is involuntary. This portion of the nerves control those internal organs of the body, and it includes such things such as, for example, when our stomach is digesting and churning its content, or the movement of our digested food through our intestines. And we are generally unaware of these types of changes, these types of happenings, and have absolutely no conscious control. These are completely involuntary. So this involuntary part of the peripheral nervous system is maintained by the sympathetic and also the parasympathetic system, which balanced each other, very similar to on and off type of switches. Looking at the nerve cell or the neuron, the nerve cell or the neuron, and here are examples of the nerve cell or the neuron. This is the functional unit of the nervous system, and there's three main types of neurons. Each of them have their own specific functions. The first type is a sensory neuron, and a sensory neuron, or another name for it, is the afferent neuron. These neurons sense and then relay the information from the environment to the central nervous system for the signal to be processed. These types of sensory neurons or afferent neurons are located in clusters or in tracts called ganglia. And these are found just outside of the spinal cord, these types of sensory neurons. Then there is the interneuron, or these association neurons that form a link within the body, and predominantly in the brain and the spinal cord. So those are the type of neurons that are in between. And the third type of neuron is the motor neuron here, or the efferent neuron is the other name for this group of nerves. And the motor neurons or the efferent neurons relay information from the brain to the effectors, such as muscles or organs or glands, in order to counteract whatever is the recognized change in the environment. But altogether, all of these three types of neurons are composed of three basic parts. There are the dendrites, and you can see them over here, these branched structures. These are the dendrites. The dendrites receive information from other nerve cells and then direct it towards the cell body. The other main feature common to most neurons is the actual cell body itself, which contains your standard set of cell organelles and carries out regular metabolic processes. And the cell body then has an extension from the cytoplasm outwards called the axon. And the axon directs nerve impulses or information, that's what the nerve impulses are, away from the cell body out towards either the what's the word I'm looking at? The effector, where the muscle or the gland, or whatever it is that the signal needs to be sent to, or to other neurons. Most of the time, these axons are covered with what is an insulating sheath that is made up of fats and proteins, and the name for this sheath is the myelin sheath. Myelin sheath is actually made by these special glial cells that are called Schwann cells, and Schwann cells help to form this myelin sheath. They kind of curl and wrap themselves around the axon, forming this myelin sheath. And the job of this myelin sheath is to help to prevent the loss of those charged ions from the nerve cell as it's going down the axon. And then there's these spaces in between as the myelin sheath goes down the axon. And these points are called the nodes of Ranvier. These breaks at the myelin sheath. And these actually help to speed up the transition of nerve impulses. In a way, it helps the signal to jump from node to node to node down the axon. So it helps to really speed up the speed at which that nerve signal moves down the body of the axon. And in the peripheral nervous system, there are additional features. These are called neurolemmas, which actually also surround the axon. These are thin membranes that allow the axons then to regenerate when they are damaged. So this is very useful in the peripheral nervous system. In the brain, there's this type of regenerative cell, which is surrounded by its myelin sheath, and this is referred to as the white matter. And the gray matter consists of unmyelinated fibers, and because of their gray color, they're called the gray matter because of that. And these are not able to regenerate themselves in response to injury. So it's only certain areas of the nervous system that once they're damaged, the function can be restored and they have the ability to repair themselves. But this process is a process that occurs very slowly, and very, very slowly, and as slow as sometimes 1 millimeter per year of regrowth in response to repair. Sometimes some people that have nerve damage in their limbs, you have that that feeling of numbness, and that very, very gradually, slowly sensation and feeling comes back after a number of years. But unfortunately, those neurons that are in the spinal cord don't have this type of regenerative ability. So oftentimes, if the nerves of the spinal cord are damaged, this leads to paralysis. Of the system. We talk about the neuron and what is its function, the sensory neuron or the afferent neuron. Then there's the interneuron and the motor neuron. So these are the three main types or classes of neurons: sensory neuron, interneuron, motor neuron. Then we also talked about the dendrite and the axon and what is their function. We talked about the myelin sheath and the nodes of Ranvier, how they are these regularly occurring gaps between sections of the myelin sheath that really help to speed up the induction of the nerve signal. We discussed neurolemma, which is that delicate membrane surrounding axons of some types of nerve cells. And we will be talking, we haven't talked about yet, but we will be talking about the reflex arc. Now let's discuss the electrochemical impulse. So when the neuron is not firing and is not relaying information, it is said to be at rest. And this is a state where there is, when it's at rest, in this situation, there's a charge difference across the membrane. And this is because there is an unequal concentration of positive ions, being sodium and potassium, on either side of the membrane. So this unequal concentration of positive ions, with more positive ions of sodium being outside of the membrane and fewer potassium being inside of the membrane, and the rate of the return of the potassium, of the sodium ion, return of the sodium ion into the membrane is much slower. And the rate of the sodium ions leaving to go outside of the neuron are much greater. So it leads to this big difference in the concentration of sodium ions outside being greater, the concentration of potassium ions inside, and this difference in charge is called the resting membrane potential. And there is a difference in a negative 17 millivolts across the membrane between the outside and the inside. So in this case, you have potassium ions that are concentrated on the inside of the cell, sodium ions are concentrated on the outside. Both of these ions can move across the membrane, but the membrane is far more permeable to the potassium. So you have more potassium diffusing out of the cell than sodium is able to diffuse in. And so because you have this this unequal movement of these ions, then that's why the exterior of the cell becomes more positive relative to the inside. And so when you have this this situation where the nerve is said to be at rest, there is said to be a polarization in the membrane. So then when the nerve impulse happens, the nerve impulse is an electrochemical message that is generated by the movement of ions through the membrane. And when the nerve gets excited, there is a potential charge difference across the membrane that changes from this minus 70 millivolts to plus 40 millivolts. And this is called the action potential. And it's shown here. See, this is the potential at rest. And then you have a nerve impulse that increases the action potential to around 40 millivolts. That is shown here in this as the direction of the nerve impulse travels down the membrane. When you have this excitation of the nerve, of the neuron, at this point, the membrane tends to become more permeable to sodium ions. And it's thought that the membrane has sodium ion channels embedded within it. And as these sodium gates open, you have, as these sodium gates open, those potassium gates close, and this allows for a very rapid flow of sodium ions into the cell, causing a charge reversal here, which is referred to as a depolarization. And once the interior of this of the nerve cell becomes positive, then the sodium and potassium gates will then reverse themselves, and you have a sodium-potassium pump that will restore that ion concentration back to its resting levels with more sodium ions concentrated outside. So sodium gets pumped back out and potassium ion pumped in. So here in this diagram, we show how the action potential travels across and down the membrane as sections become depolarized and then repolarized again due to the action of the sodium-potassium pump. So then the nerve cell cannot send another impulse again until this membrane has been restored or repolarized back into its resting state. And this period that it takes in order for this, this repolarization to be established, this is called the refractory period, and sometimes it can last anywhere from 1 to 10 milliseconds. So this wave of depolarization, as it sweeps down the length of the axon in sections, you can think of it very similar to when you're in the stadium and you have the crowd doing a wave. So there's this wave of depolarization that that works its way, sweeps its way down the length of the axon. And behind it, you have the repolarization as the concentrations of sodium and potassium ions are then again restored back to their resting state. So what will then cause this excitation in the neuron? So when you have a sensory neuron that detects a change in the environment, and this change we call a stimulus, this stimulus that can be detected by that sensory neuron, it has to be of a strong enough magnitude to trigger that depolarization of the membrane. And the intensity of that stimulus has to reach a certain set level or a threshold level in order for the signal to be sent. And it's very important for there to be a threshold level established, because it just then stops small changes that really don't have much of an effect from really sending signals to the brain. And without there being that threshold level established, you can imagine that those sensory neurons would just be continuously firing and sending signals and overwhelm the brain. So it's important for there to be a certain threshold, so for the stimulus to be strong enough to trigger excitation of the neuron. So once that threshold level is reached, then the neuron will fire at the same intensity and at the same speed for any stimuli. And the size of the charge can't affect these factors of the nerve impulse. So if you have a greater stimulus, it doesn't necessarily cause a greater depolarization of the membrane. This is called the all or none response. And in this case, the neuron will either send a signal or it does not. So it's an all or none. Then how does the brain then interpret whether or not there is a signal of different strengths? For example, how does the brain know the difference between when you have stubbed your toe and it hurts only a little bit, or you've stubbed your toe a lot and you've hit it so hard that it hurts a lot? How does the brain distinguish between that? That happens when the brain is able to interpret the intensity indicated by the frequency of the nerve impulses being sent, how quickly, how fast those nerve impulses are arriving. So therefore, if you have a stimulus that's very small, only a few signals will be sent, one after another. But if a stimulus is very intense, that signal will be sent repeatedly of a very high frequency. As soon as the membrane has repolarized, it is sent again. So we have a couple of videos that we can watch together. And we'll watch the first one. On the nerve impulse.
A nerve impulse is an electrical current that travels along dendrites or axons due to ions moving through voltage-gated channels in the neuron's plasma membrane. Voltage-gated channels open and close in response to an electrical voltage, so they are affected by changes in electrical charge around them. When a neuron is at rest, a charge difference is maintained between the inside and outside of the cell. This charge difference is produced and maintained largely by active transport using sodium-potassium pumps. The pumps send sodium ions out of the cell and bring potassium ions in. While other channels allow some flow of potassium ions back out of the cell, the sodium ions cannot easily get back in to replace the lost positive charges. The overall result is that the exterior of the cell has a net positive charge and the interior has a net negative charge. The difference in charge between the interior and exterior of the cell is called the resting membrane potential. A nerve impulse begins when a stimulus disturbs the plasma membrane on a dendrite, causing sodium channels to open. Sodium ions flow into the cell, lessening the charge difference at that location.
So these channels are different from that sodium-potassium pump that differentially pumps sodium ions to the outside and potassium ions to the inside. That's a sodium-potassium pump. It requires an input of energy. It requires ATP in order to pump sodium ions outside, pump potassium ions inside, therefore establishing a difference between the two. And because you have more sodium outside, then you have potassium inside, there seems to be an apparent difference in the polarity, so that outside is more positive compared to inside. Relatively, the inside is negative and the outside might be positive in relation to the outside. These are channels which are specific to each ion, and once they open, they allow the flow of these ions back inside or outside of the cell.
If the change is enough, it will cause nearby voltage-gated sodium channels to open. This allows so many sodium ions to flood into the cell at that location that the membrane there is depolarized, with the local region inside the cell having a net positive charge and the outside of the cell having a net negative charge. This affects neighboring voltage-gated sodium channels, which then open, moving the depolarization along the membrane. This moving depolarization is called an action potential. Changes occur behind the action potential to restore the resting membrane potential. The voltage-gated sodium channels close and voltage-gated potassium channels open. This allows a rapid flow of potassium ions out of the cell, repolarizing the membrane so that the inside is again negative and the outside positive. This is followed by use of sodium-potassium pumps to fully restore the resting membrane potential and to reestablish proper concentrations of sodium and potassium ions inside and outside the cell. A nerve impulse is an electrical.
Okay. So that was an example of a nerve impulse being transmitted down the axon electrically. There's also electrochemical transmission or synaptic transmission. So if you look at the distance between what is our peripheral nervous system and the brain, so if you think of, for example, the distance between the tip of your finger and your brain, or your feet and your brain, this distance is really too great for a single neuron to stretch that entire distance. So the information from the sensory neurons that are in your fingertips or are in your feet have to be passed to interneurons, which will then pass it again along to the nerves of the brain. This means that when the signal reaches the end of your sensory neuron, there has to be some mechanism in place to transfer that signal to the interneuron, and then from the interneuron to the neurons of the central nervous system. And this mechanism happens through synaptic transmission. In synaptic transmission, there is a very, very small space between the ends of the neurons called a synapse. And when the signal reaches the end of the neuron, you have chemical neurotransmitters that move across this space, also called the synaptic cleft. This space between is called the synaptic cleft. And these small molecules, which are held in very, very tiny vesicles at the end of the first neuron, these are called the presynaptic neurons because they exist just before the synapse, so they're the presynaptic vesicles. These vesicles move to the end of the neuron. They will fuse together with the membrane at the end where the synaptic cleft is. Let me just zoom in for you. And this is a picture, a diagram that I've got directly from your grade 12 Nelson biology textbook. So these presynaptic vesicles will fuse with the membrane here and really deposit or empty their contents into this space, into this gap, into this synaptic cleft. Then this next neuron here, this will, this one is called the postsynaptic neuron. So the pre and the post, the one before bringing the signal and the one after, which will be receiving the signal. This has got these receptors, these little red colored receptors on its end, and these receptors will recognize and bind these neurotransmitters that are released from the presynaptic vesicles. And once they are bound to these receptors, a threshold level is reached, which then allows for the signal to move down the length of the neuron and travel once again down the axon. So it is this diffusion of these transmitters across the cleft that slows down the transmission of the signal, so that you need a greater number of synapses and but a slower traveling of the signal. And one of the neurotransmitters called acetylcholine, a very common neurotransmitter, that is able to once it travels across the synaptic cleft and binds to its receptor, it opens the sodium channels in that postsynaptic neuron. And once that message gets transmitted, then you have another enzyme called choline esterase that gets released back into the synaptic cleft here to break down any extra acetylcholine that may be left behind there, to break down the acetylcholine to then end that signal. There's another video that we can watch together. That's here. And there's some information before it, and you can watch it if you like on your own time, but I just kind of moved it ahead to the spot where really our relevant information is.
Nerve cells have two main settings for communicating with each other, depending on how fast the news needs to travel. Some of your synapses are electrical. That would be like an immediate group text. Others are chemical synapses. They take more time to be received and read, but they're used more often and are much easier to control, sending signals to only certain recipients. Fortunately, your nervous system has better text etiquette than your mom and knows when each kind is appropriate to use and do it. Your super fast electrical synapses send an ion current flowing directly from the cytoplasm of one nerve cell to another through small windows called gap junctions. They're super fast because the signal is never converted from its pure electrical state to any other kind of signal the way it is in a chemical synapse. Instead, one cell and one synapse can trigger thousands of other cells that can all act in synchrony. Something similar happens in the muscle cells of your heart, where speed and team effort between cells is crucial. This seems like a good system, so why aren't all of our synapses electrical? It's largely a matter of control. With such a direct connection between cells, an action potential in one neuron will generate an action potential in the other cells across the synapse. That's great in places like your heart, because you definitely don't want half a heartbeat. But if every synapse in your body activated all of the neurons around it, your nervous system would basically always be in group text mode, with every muscle fiber and bit of organ tissue always being stimulated and then replying all to the whole group, which would stimulate them even more until everyone just got maxed out and exhausted and turned off their phones for good, which would be death. So that would be bad, which is partly why we have chemical synapses. They are much more abundant, but also slower, and they're more precise and selective in what messages they send where. Rather than raw electricity, these synapses use neurotransmitters, or chemical signals that diffuse across a synaptic gap to deliver their message. The main advantage chemical synapses have over electrical ones is that they can effectively convert the signal in steps, from electrical to chemical back to electrical, which allows for different ways to control that impulse. At the synapse, that signal can be modified, amplified, inhibited, or split, either immediately or over longer periods of time. This setup has two principal parts. The cell that's sending the signal is the presynaptic neuron, and it transmits through a knob-like structure called the presynaptic terminal, usually the axon terminal. This terminal holds a whole bunch of tiny synaptic vesicle sacs, each loaded with thousands of molecules of a given neurotransmitter. The receiving cell, meanwhile, is, yes, thankfully, the postsynaptic neuron, and it accepts the neurotransmitters in its receptor region, which is usually on the dendrite, or just on the cell body itself. And these two neurons communicate even though they never actually touch. Instead, there's a tiny gap called a synaptic cleft between them, less than 5 millionths of a centimeter apart. One thing to remember is that messages that travel via chemical synapses are technically not transmitted directly between neurons like they are in electrical synapses. Instead, there's a whole chemical event that involves the release, diffusion, and reception of neurotransmitters in order to transmit signals. And this all happens in a specific and important chain of events. When an action potential races along the axon of a neuron, activating sodium and potassium channels in a wave, it eventually comes down to the presynaptic terminal and activates the voltage-gated calcium channels there to open and release the calcium into the neuron's cytoplasm. This flow of positively charged calcium ions causes all those tiny synaptic vesicles to fuse with the cell membrane and purge their chemical messengers. And it's these neurotransmitters that act like couriers, diffusing across the synaptic gap and binding to receptor sites on the postsynaptic neuron. So the first neuron has managed to convert the electrical signal into a chemical one, but in order order for it to become an action potential again in the receiving neuron, it has to be converted back to electrical. And that happens once a neurotransmitter binds to a receptor, because that's what causes the ion channel to open. And depending on which particular neurotransmitter binds to which receptor, the neuron might either get excited or inhibited. The neurotransmitter tells it what to do. Excitatory neurotransmitters depolarize the postsynaptic neuron by making the inside of it more positive and bringing it closer to its action potential threshold, making it more likely to fire that message onto the next neuron. But an inhibitory neurotransmitter hyperpolarizes the postsynaptic neuron by making the inside more negative, driving its charge down, away from its threshold. So not only does the message not get passed along, it's now even harder to excite that portion of the neuron. Keep in mind here, any region of a single neuron may have hundreds of synapses, each with different inhibitory or excitatory neurotransmitters. So the likelihood of that postsynaptic neuron developing an action potential depends on the sum of all of the excitations and inhibitions in that area. And we have over 100 different kinds of naturally occurring neurotransmitters in our bodies that serve different functions. They help us move around, keep our vital organs humming along, amp us up, calm us down, make us hungry, sleepy, or more alert, or simply just make us feel good. But neurotransmitters don't stay bonded to their receptors for more than a few milliseconds. After they deliver their message, they just sort of pop back out and then either degrade or get recycled. Some kinds diffuse back across the synapse and are immediately reabsorbed by the sending neuron in a process called reuptake. Others are broken down by enzymes in the synaptic cleft or sent away from the synapse by diffusion. And this mechanism is what many drugs, both legal and illegal, so successfully exploit in order to create their desired effects. These drugs can either excite or inhibit the production, release, and reuptake of neurotransmitters. And sometimes they can simply mimic neurotransmitters, tricking the neuron into thinking it's getting a natural chemical signal when really it's anything but. Take cocaine, for example. Don't take cocaine. Once it hits your bloodstream, it targets three major neurotransmitters: serotonin, dopamine, and norepinephrine. Serotonin is mainly inhibitory and plays an important role in regulating mood, appetite, circadian rhythm, and sleep. Some antidepressants can help stabilize mood by stabilizing serotonin levels. And when you engage in pleasurable activities like hugging a loved one or having sex or eating a really great donut, your brain releases dopamine, which influences emotion and attention, but mostly just makes you feel awesome. Finally, norepinephrine amps you up by triggering your fight or flight response, increasing your heart rate, and priming muscles to engage, while an undersupply of the chemical can depress a mood. So in a normal, sober state, you got all these neurotransmitters doing their thing in your body. But once they've delivered their chemical payloads, they're usually diffused right back out across the synapse to be absorbed by the neuron that sent them. But cocaine blocks that reuptake, especially of dopamine, allowing these powerful chemicals to float around and accumulate, making the user feel euphoric for a time, but also paranoid and jittery. And because you have a limited supply of these neurotransmitters and your body needs time to brew more, flooding your synapses like this eventually depletes your supply, making you feel terrible in a number of ways. Cocaine and other drugs that target neurotransmitters trick the brain, and after prolonged use may eventually cause it to adapt, as all of those synapses remember how great those extra chemicals feel. As a result, you actually start to lose receptors. So it takes even more dopamine and finally cocaine to function normally. Sometimes the best way to understand how your body works is to look at how things can go wrong. And when it comes to your synapses, that, my friends, is what wrong looks like. In their natural healthy state, your synapses know when to excite, when to inhibit, when to use electricity, and when to dispatch the chemical messengers. Basically, a healthy nervous system has the etiquette of electrical messaging down to, well, science. Today, you learned how electrical synapses use ion currents over gap junctions to transmit neurological.
Right. Which now brings us to a discussion of the reflex arc. So in most situations, let me just zoom here for you. Here we go. So in this diagram shows what we mean when we discuss the reflex arc. So in most situations, you have the nervous system working quickly enough to respond to those changes in the environment. But then you have other situations where you need an almost instantaneous type of response in order to keep you out of harm. This is the function of the reflex arc. And each reflex arc contains 5 necessary components. Where am I? There I am. One being the the sensory receptor, which will sense, for example, the stimulus. In this case, the sensory receptor is at the end of the finger, will sense the sharp, pointy tip of this tack as the stimulus. Then you have the sensory neuron, which will send the signal directly towards in the direction of the brain. And once the signal then reaches the spinal cord here, there's specialized interneurons here that have pre-programmed function to have a specific response. And their response then gets sent directly back through the motor neuron, back to the effector, which is the muscles here, and causes the muscles to contract. And at the same time, the signal also travels towards the brain for processing, so that by doing this reflex arc and processing the signal immediately in the spinal cord, even before it is sent to the brain and perceived by the brain, this cuts down that lag time that you need in order for the signal to reach the brain, have the brain process then send the appropriate response. So in this case, the reflex arc causes the muscle to contract and for your hand to pull away your finger from the sharp object, even before your brain has fully perceived that you are touching a sharp object. Another example of this is when, for example, you might be touching something hot. In that case, the reflex arc causes you to pull your hand away before you seriously damage your hand and your finger. And it's only after you've pulled back your finger that you actually then perceive that feeling of heat. So in this case, the reaction comes first and then the sensation. So that is one way. There's this protective mechanism that the reflex arc provides. And we can also watch a video on the reflex arc together. And I have that one queued up here.
Rare, the rest of us have a whole nervous system dedicated to making sure our bodies react with a predictable chain of events at the first sign of damage. Like say you just wake up and you're extraordinarily hungry for some reason, so you run downstairs to grab some clam chowder, but you didn't put any shoes on, and suddenly you're like, yeah! An attack fell out of the wall and you stepped right on it, of course. Your foot immediately lifts off the ground, and then you're assuring your dog that you're not yelling at her, you're just yelling, and then you limp over to the couch and sit down and pull up your foot and remove the spiny devil from your flesh. You want to talk physiology? So what exactly just happened in your body? Well, the first step was a change in your environment. That is, a stimulus that activated some of your sensory receptors. In this case, it was a change from the probably completely ignored feeling of bare skin on a smooth floor to a distinct feeling of discomfort, the sharp metal tack piercing your skin. Your peripheral nervous system's mechano and nociceptors provided that base sensation or awareness that something had changed. Then it went to your central nervous system, first to the spinal cord that caused the immediate reflexive action of pulling up your foot, and then your brain eventually interpreted that awareness into the perception of pain and decided to pull the tack out and probably say an expletive or two. Pain itself is a pretty subjective feeling, but the fact is, we all have the same pain threshold. That is, the point where a stimulus is intense enough to trigger action potentials in those nociceptors is the same for everybody. But you and I might have different tolerances for discomfort. In general, most doctors think of pain as the perception, whatever any given brain says pain is. So you got the stimulating event, foot meets tack, and then you got the reception of that signal as the nociceptors in your foot sense that stimulus, and then the transmission of that signal through your nerves to your spinal cord and eventually up to the brain. Now, remember back how every neuron in your body has a membrane that keeps positive and negative charges separated across its boundaries, like a battery sitting around waiting for something to happen. Well, that tack in your flesh is that something, and it snaps those nociceptors to attention. Some neurons have mechanically gated receptors that respond to a stretch in their membranes. In this case, that happens when the tack punches through them. Meanwhile, other neurons have ligand-gated receptors that open when the damaged skin tissue releases chemicals like histamine or potassium. These channels allow sodium ions to flood into the neuron, causing a graded potential. If that hits the right threshold, it activates the electrical event that sends the signal all the way up the axon and gets one neuron top into another, the action potential. When that action potential races down the length of its axon to the terminal, the message hits the synapse that then flings it over that synaptic gap to another neuron that's in your spinal cord. Remember, signals travel between neurons either by electrical or chemical synapses. The electrical ones send an electrical impulse, while the chemical ones, the one we're talking about right now, first convert that signal from electrical to chemical by activating neurotransmitters to bridge the synaptic gap before the receiving neuron converts that chemical signal back into an electrical one. In this case, news of the tac attack is carried by specific neurotransmitters whose sole job is to pass along pain messages. So far, your body's response to the stimulus has been handled by the sensory or afferent division of your peripheral nervous system. This is the part that's involved expressly in collecting data and sending it to the central nervous system. But at this point, the responsibility changes hands. The torch is passed because the pain signal has just triggered an action potential in a neuron in the spinal cord, which is part of the central nervous system, and there it reaches an integration center. From here, the response is taken over by the motor or efferent division. Once the integration center interprets the signal, it transmits the message to motor neurons, which send an action potential back down your leg where it reaches an effector. And an effector is just any structure that receives and reacts to a motor neuron signal, like a muscle contracting or a gland secreting a hormone. From here, the motor neurons complete the whole foot lifting response until the rest of your nervous system gets engaged in the complicated tasks of figuring out what the problem is and fixing it. Those are the 5 steps that your highly specific neural pathways go through to produce what's known as a reflex arc. A lot of your body's control systems boil down to reflexes just like this. Immediate reactions that can either be innate or learned, but don't need much conscious processing in the brain. Lifting your foot when you step on a tack is an innate or intrinsic reflex action, a super fast motor response to a startling stimulus. These reflexes are so invested in your self-preservation that you actually can't think about them before you respond. This processing happens in the spinal cord so that the control of the muscles can be initiated before the pain is actually perceived by the brain. Learned or acquired reflexes, on the other hand, come from experience, like how you learn to dodge obstacles while riding a bike or driving a car. That process is also largely automatic, but you learn those reflexes by spending time behind the wheel or behind the handlebars. And reflex arcs stimulate some muscles while inhibiting others. For example, the tack in your right foot ended up activating the motor neurons in your right hip flexors and hamstring, causing your knee to bend and your foot to lift up. But it also told the quad muscles in your left leg to extend and stand tall, allowing you to shift your body's weight off the tack. Of course, not all reflexes come from pain, as you've probably experienced when a doctor tapped your knee and foot kick. Your muscles and tendons are very sensitive to being stretched too far or too fast because that kind of movement can cause injury. So for this, we have receptors called muscle and tendon spindles that specifically sense stretching. If triggered by an overstretch, they generate a reflex arc that contracts the muscle to keep it from stretching further. So when does the brain actually get involved in all this? Well, when your spinal cord sent impulses down the motor neurons, it also sent signals up your spinal cord toward the brain. News of the attack arrived first at your thalamus, the information switchboard that then split the message and sent it to the somatosensory cortex, which identifies and localizes the pain, like sharp at foot, as well as the limbic system, which registers emotional suffering, like why tack, why knee? And it also went to the frontal cortex, which made sense of it all, assigning meaning to the pain, like, oh, I see, this tack fell to the crash course poster on the wall here. So basically, although your body has been reacting all along, it's not until those pain signals hit the brain that you have the conscious thoughts of both, dang, that hurt, and oh, that hurt because I stepped on a specific pointy thing. And this is where I want to point out that we here at Crash Course cannot be held responsible for any injuries sustained in the process of owning a Crash Course poster.
All right. So that was the arc reflex and how the arc reflex helps to prevent injury. Now, the central nervous system, which we have been talking about, consists of both the brain and the spinal cord. And that's where you have that coordination center for all of the activities that is perceived by our nervous system. This system is very well protected because of its importance by that tough bony membrane, our skull. And underneath that, there are 3 layers of membrane, and these are called the meninges, and they serve to, they act as a protective barrier. You have the outer membrane, which is the toughest one, called the dura mater. Underneath that outer membrane, there is a mid layer called the arachnoid membrane, and then there is the innermost membrane called the pia membrane. Together, these 3 layers of membranes, these meninges, form the blood-brain barrier, which limits what type of substance can move from the bloodstream into the nerves, into the central nervous system. In between that middle and inner layer, that's where you have the cerebrospinal fluid. And the cerebrospinal fluid acts as a shock absorber, as a way of transporting nutrients to the nerve cells, and also as a way of removing waste and returning the waste back to the bloodstream for disposal. The spinal cord portion of the central nervous system, that's the part that carries information from the peripheral nervous system to the brain for processing. So the spinal cord extends all the way down from the brain, which would be somewhere up here, through this canal that is in each one of these bones, these vertebrae. And as we talked about here, both myelinated and unmyelinated fibers then travel through the cord in these specific tracts that bring sensory information into the cord, while you have the ventral nerve tract carrying motor information from the brain out to the periphery. Now, in the brain, where you have great complexity that is necessary to establish the difference really between what we can do as humans and what other animals might be capable of. But overall, the structure of the brain between most animals is kind of relatively the same. You can divide the brain into 3 distinct regions. You have the forebrain, the midbrain, and the hindbrain. In humans, our forebrain is our largest, is the largest section of our brain. That's where we have our olfactory lobe, which processes information about smelling. That's where we also have the cerebellum, which is also located here. Sorry, the cerebrum. And that's the part that coordinates that sensory information. And in humans, also speech and reasoning and memory, these all are found in this area, in the cerebrum, in the forebrain. The surface of the cerebrum is known as the cerebral cortex. It is composed of the gray matter that is highly folded. You can see all the folds here on the surface of the brain. And these folds are meant to increase that surface area of the brain. And then you have each half of this part of the brain storing different kinds of information. On the right side of the brain, that is associated with visual and spatial awareness, while you have the left side of the brain linked to more verbal skills. And then the two sides are really able to interact through a bridge that connects the two halves. And that bridge is called the corpus callosum. I don't have a picture of it here, but there is a connection in the middle between the two halves of the brain. And then this part of the cerebrum can then be further subdivided into the different lobes, each of them having its own specific function. You have the frontal lobe, you have the parietal lobe here, you have the occipital lobe, and the temporal lobe. And down here in this chart, we have a summary of the different functions associated with each one of these lobes, so that the frontal lobe, as we said, has to do with control of the movements, aspects of memory, and also inhibition of unsuitable behaviors. The temporal lobe, here in purple, this purple region, is the sensory areas associated with vision and hearing, and also link these vision and hearing associations that are linked specifically to memory and the interpretation of that type of sensory information. Then in the parietal lobe, which is this yellow region here of the brain, that is associated with touch and temperature awareness, and also interpreting pain and our direction of orientation. And it's also been linked to emotions and interpretation of speech. And finally, the occipital lobe over here, this green shaded region, that's the sensory areas associated with vision and the interpretation of visual information. So then the area of the midbrain has these 4 spheres of gray matter that can act to relay information as a relay center for relaying information from both the eyes and the ears. In the hindbrain, which is found posterior to the midbrain, this joins with the spinal cord, and the hindbrain consists of the major regions of the cerebellum, the pons, and also the medulla oblongata. Here, the cerebellum is that largest section that is found immediately behind this cerebrum region. It controls limb movement, balance, and also muscle tone. The pons acts as a relay station that passes information between the regions of the cerebellum and between the cerebellum and also the medulla oblongata. And then this last section, the medulla oblongata, acts as the connection between the peripheral nervous system, which would be down here past the spinal cord, and the central nervous system, which would be the spine and the brain. It also is in control of involuntary muscle action, and can regulate things such as the breathing and the movement necessary for breathing. It can control the diameter of our blood vessels, as well as the heart rate, and also acting as the coordinating center for the autonomic nervous system. So this structure and the function of the brain is really a great mystery that is still being worked on and teased apart. And there was a Canadian physician called Wilder Penfield, and he's considered to be the original pioneer in brain mapping and figuring out what different parts of the brain actually do. And he used electrodes to actually electrically stimulate different areas of the brain to determine what is their function. And his focus was on mapping the cerebral cortex, and he used epilepsy patients to do this. When a patient has epilepsy, they have massive electrical misfiring of their neurons within their brain. And so he was able to use this to determine the area where the problem originated. So, and by doing that, tissue could be removed from that area, leaving rest of the cortex intact. Quite incredible. So then there is the autonomic nervous system. If you remember, we talked about the autonomic system that worked together with the endocrine system to really adjust those, the internal environment. And this entire system of the autonomic nervous system working together with the endocrine system really works without us having any conscious control over it. In place are those two antagonistic portions of the system that work to maintain that balance. Antagonistic meaning working against each other. So you have the sympathetic nervous system that prepares the body for stress, while you have the parasympathetic nervous system that restores back into the body back to normal function. And so here in this table, we show the effects of both of these systems, the sympathetic and the parasympathetic, on the various organs of the body. So if we look at the heart as one of the organs, one of the major organs, the effect of the sympathetic nervous system is to prepare the body for stress, and it does so by increasing the heart rate. And of course, once the stress is gone, the parasympathetic nervous system returns and restores the function back to normal and decreases the heart rate to come back down to its normal level. Another example is if we look at the digestive organs, the sympathetic nervous system in response to stress will decrease the peristaltic motions of the digestive system, essentially not using up the resources for digestion of food at a time of stress. While the parasympathetic system, once the stress is gone, will increase peristaltic motion in order to carry out all the digestive processes again. The liver is another major organ, and the sympathetic nervous system in preparing the body for stress will act to increase the release of glucose from the liver. Again, by releasing glucose, it makes it available as an energy source to the muscles and cells of the body as they are preparing to react to the stress. And then antagonistically working against it, once that stress is gone, the parasympathetic system then restores glucose stores within the liver. Looking at the eyes, the sympathetic nervous system will work to dilate the pupils and make them larger, allowing more entry of light, so to enhance vision and perception. while parasympathetic nervous system, once the stress is gone, will constrict the pupils back down to their normal size. The bladder in respect to the sympathetic system might relax its sphincter, perhaps to discharge the content, preparing for that fight or flight response, while the parasympathetic will contract the sphincter back to its normal state. The skin, in response to the stress, sympathetic nervous system will increase blood flow to the skin, while the parasympathetic nervous system will decrease blood flow and return blood flow in the skin back down to its normal levels. The other major gland, the adrenal gland, if you recall, it has the ability to release epinephrine, and the sympathetic nervous system causes the release of epinephrine from the adrenal gland, while the parasympathetic system will have no effect, and eventually the levels of epinephrine will return back down to normal. So if we look at the sensory receptors that we talked about, we can perceive and interpret various stimuli from the outside world using our special sensory receptors. These specially modified ends of our sensory neurons, each one of them is able to respond to a particular type of stimulus, and the information that is provided by these stimulus allows our brain to then respond appropriately to this sensation and to maintain either homeostasis or act to prevent damage to us as the organism. And for some types of stimuli, we respond very instinctively, while there's other types of responses that have to be learned. So here in this chart, we can look at the different types of receptors and the type of information that they provide to the brain. For example, our taste buds, our taste receptors will respond to chemical stimulus, and the type of information is, for example, the presence of a specific chemical that will have a certain taste, for example, sweetness or sourness, and they will identify the presence of these specific chemicals by our taste buds and provide that information to the brain. Our smell receptors, again, will respond to chemical stimuli and will provide the presence of chemicals as detected by our olfactory cells and the smell receptors to different parts of the brain. Our pressure receptors will respond to mechanical stimuli that will provide either pressure or a sense of pain, and they will provide information as to the movement of the skin or changes in the surface of our body in response, for example, touch. There are proprioceptors. They will respond to a mechanical movement, for example, movement of the limbs. There are balance receptor inside our inner ear, which will respond to mechanical stimulus and body movement. There is receptors in the outer ear that will respond to sound and sound waves. The eye will respond to light stimulus and changes in light intensity and movement and color, for example. And finally, thermal regulators, which will respond to heat and provide information about the flow of heat and that can detect heat or lack thereof it. So for us, taste and smell really go together in order to help us identify specific tastes of food. And the majority of what we actually taste when we are tasting something is due to the smell of food. So if we've ever had a cold, for example, as I think many of us may have had recently, we've realized that when our noses are stuffed up, we can't taste food, or at least we can't taste very well. Then we have proprioceptors, which respond to stretching and relaxation of muscles. And even with our eyes closed, we are actually quite aware of the position of our limbs due to the information that we receive from what muscles are relaxed and which muscles are contracted. And this is something that sometimes has to be learned. But for example, in babies, they don't have very much conscious control over their limb movement. So it's only through repetition as they learn these movements that they learn how to control their limbs and gain control over their limbs. Our balance, our sense of our balance is something that's controlled by our inner ear. In our inner ear, we have these semicircular canals that contain fluid that moves around based on the position of our body. And within them, there are these tiny hairs that are able to sense the movement of the fluid inside the canal and send that information to the brain. And then the brain is able to interpret that information from the sensors to determine if the individual is standing upright or leaning over, for example. So if you, for example, move and spin in a circle for a few moments and then stop, you have this feeling that the room is spinning. And this is because fluid keeps moving within your ear canals, even still after a few moments after you have stopped spinning. That fluid keeps on moving and sends that information to your brain, and then your brain interprets this lingering movement of the fluid to mean that your body is still moving, even though you have stopped. Then the touch sensors in our body, which are extremely concentrated, especially in some areas, such as our fingertips, and less concentrated in others. That simply depends on on the sensitivity that is required in that area and how important it is for that area of the body to be very sensitive or only slightly sensitive. Our fingertips have high concentrations of these touch receptors, as does, for example, our neck region. And because we use our fingertips to do so many tasks and even investigate our external environment for that sense of touch, the sensations coming from our fingertips are very important. In the neck region, because this area is the area that contains the spinal cord, therefore it's important for it to have this increased sensitivity for you to have that feeling of wanting to have that protection. So both our ears and our eyes are very special sensors that provide information to us, to our organism, and hearing then allows us to gather information about the environment from a distance, from far away, as kind of an early warning system. We would be able to detect from even far away what's coming. And the eyes then allow us to perceive objects that might be present from a distance, and also to closely examine objects that are closer to us and are very near. And this system, this visual system, the eyes react to light stimulus. And for protection, our eye has the ability to adjust the amount of light that can enter the eye by controlling the dilation and the contraction of our pupil. And finally, our body is able to sense heat and cold in order to prevent damage to the organism. And that brings us to the end of our lesson. On the nervous system. Does anybody have any questions?